Thank you, Bob. I'm really delighted to be here, not only because I got to go to Mississippi and see my mom, which is always a good thing, but because I finally get to spend some time here and see the things you're doing, about which I've heard for a number of years. I have a former undergraduate student who's in my lab, who's at Emory now, and he waxes eloquent about both Emory and Georgia Tech, so it's nice to actually be here. I'm also a guinea pig at using this fancy newfangled device, so bear with me if I don't get it exactly right. I'm not an engineer, I'll say that up front. I really want to spend some time telling you what we've worked on for the last literally almost 10 years, and something about which I really pose the question, cell transplantation for myocardial repair, are we ready for prime time? And the reason I ask that is that although we've been working on this technology for about the last 10 years, even as recently as 3 years ago, no one was really talking about this. We couldn't even get people to let us talk about this at American Heart or at a number of other meetings. And now, a mere 3 years later, there are clinical trials popping up everywhere in this technology. And so the question is, are we moving ahead too rapidly, or are we moving ahead in a way that makes sense for the field? And I hope you'll join me in exploring that question. So what I really want to do is point out, first of all, that it is already.
Fortunately, you're Steve.
That's right. Steve is going to fix my mistakes, but not. So it is a growing field. In 1998, there were virtually, there were less than 20 publications in the field. But last year alone, there were over 100 papers published that had to do with cell transplantation into the myocardium. And already this year, I've reviewed a number of papers, and I'm sure there are many more out there about which I know nothing. So it's a rapidly growing field, and everyone's sort of jumped on this bandwagon, and I really want to explore the whole field. So I'm going to go through the concept, really give you a preclinical summary, and then talk about what current clinical trials exist and what we think the next few years look like. So this is going to be a combined science and a little bit of an introduction to clinical trials talk. So obviously, the reason that we're talking about this is because acute myocardial infarction is a huge healthcare problem in the U.S. It affects approximately 3.5 million patients every year in the U.S. And if we're going to repair the damage that occurs in the myocardium secondary to a blocked coronary artery, we're going to have to not only restore blood flow, we're going to have to repair the injured tissue. So I tend to think about acute myocardial infarction as an initiating cause that leads to left ventricular dysfunction, and as a cascade. It's a progressive disorder. And it really, if you think about what's going on, you have ischemia in the myocardium, cardiocytes die, you're trying to do more work with fewer cells, you begin to get remodeling, hypertrophy of the cells, they become less functional, and it's a vicious cycle. So what we decided to do was really try to decide what it was going to take to block that cycle. And I think it's going to take providing more cells. So we can use these cells then to either mediate functional repair of the myocardium, to promote angiogenesis, or perhaps to deliver potentially therapeutic genes to the myocardium. And I'll talk a little bit about at least the first two. So this is a very straightforward, not simple, but straightforward concept. We've focused on rats. We've focused on deriving myoblasts from a skeletal muscle biopsy, these individual cells that exist at the periphery of the muscle, expanding these cells in vitro, and delivering the cells to the injured region of myocardium. I would submit that the type of repair that you're trying to do depends upon the injury. And that if you're trying to deal with hibernating myocardium, or myocardium where The cells are not dead, they've changed their functional status and are no longer contracting due to decreased blood supply, that angiogenesis may be sufficient. But if you're dealing with an infarct where you've actually got a scar, obviously you want functional repair. But one without the other is not likely to be beneficial. That is, if you promote angiogenesis in the absence of a target, which I think is the goal for many of the current gene therapy trials with VEGF and with FGF, you really are not likely to repair contractility in the myocardium. You need a target for those cells. Likewise, if you try to put in huge numbers of cells without promoting angiogenesis, you're really not going to get good functional repair because those cells obviously need nutrients. If you're trying to repair hibernating myocardium, I submit that myoblasts plus or minus growth factors may be sufficient. And I'll talk a little bit about this. In addition, growth factors alone may work. Bone marrow-derived stromal cells may be sufficient, and at least in Europe, and now actually in the US, there's a bone marrow clinical trial that's been initiated to simply look at transplanting raw bone marrow and promoting angiogenesis in the myocardium. For functional repair, you obviously need contractile cells. We've focused primarily on skeletal myoblasts. You might ask why cardiocytes or bone marrow-derived stromal cells are not a good target, and I'm going to talk about that briefly. Again, the goal is contractile cells, so what contractile cells could you use? Why not simply induce native cardiocyte proliferation? Well, as many of you probably already know, the main reason not to do that is that we don't know how yet. Cardiocytes actually can be induced to divide their nucleus to replicate their DNA, but Michael Schneider and a number of groups have spent many, many years trying to get cardiocytes to undergo cytokinesis, and there seems to be a block. We simply can't get them to divide. The cardiocytes that we can get to divide are engineered cells, and those cells, in fact, are tumorigenic. So when we can get them to divide, we can't get them to stop. The other reason that many people don't talk about and don't think about is a much more physiologic reason, and that is that you're dealing with an infarcted region of myocardium. So if you're trying to transplant cells into that region of the myocardium and you put cardiocytes in there, 99.9% of those cells are going to die, secondary to ischemia. So you're really essentially shooting yourself in the foot to try to put new cardiocytes in that region, at least at this point. So instead, we've chosen to repopulate the scar tissue by exogenously supplied contractile cells. And again, there are a number of types of contractile cells to consider. We don't choose cardiocytes for the reasons I've already outlined. There are cells that are now known to be heart-derived myofibroblasts that were isolated in Canada by Dick Wiesel's group. And those cells may provide some benefit, but they don't seem to mediate functional repair to the extent that other cells do. We've really focused on skeletal myoblasts, also known as satellite cells, although the cells we use are not strictly satellite cells. And then I'm going to compare those cells to bone marrow-derived cells. And then obviously another option is embryonic stem cells, although at this point, in animal models, if we transplant even embryonic stem cells that are 99.9% pure, you still get tumors. So the 0.1% cells that we can't define seem to be enough to cause tumorigenicity. So those cells, we just don't understand enough about those cells yet to really use them as a cell source in these models. As I said, I'm going to compare myoblasts with bone marrow-derived cells. This is simply a bone marrow smear derived from rabbit. These are human myoblasts grown in culture. And if you look at some of the options, obviously you can derive myoblasts from an autologous source. Virtually everybody, even 80-year-old women, have a gram of skeletal muscle that they're willing to use to repair their myocardium. In bone marrow, for bone marrow, obviously, in theory, you can get enough bone marrow, although it depends on what your target cell really is and whether or not you believe you need a subset of restricted population of cells to be able to repopulate the myocardium. The myoblasts are myogenic. They form muscle naturally. Bone marrow-derived cells will form muscle. They will form myocardium if you transplant them into normal myocardium. The real problem at this point with bone marrow-derived cells is that when you transplant them into scarred regions of myocardium, by their very nature, they respond to the environment in which they find themselves, and they tend to form fibroblasts or osteoblasts or adipocytes. So they tend not to form myocardium or muscle cells in the scarred region of myocardium. And obviously, by using bone marrow or myoblasts, you don't deal with the ethical concerns associated with fetal cardiocytes or with embryonic derived stem cells, neither of which I believe are going to be good options in the U.S. for at least some period of time. Again, we've been over these reasons to use myoblasts. In addition, one of the more scientific reasons to consider using myoblasts versus bone marrow-derived stromal cells is that myoblasts normally exist in a region of skeletal muscle where their blood supply is about 1/4 of what it is in the heart. Well, in an infarcted region of myocardium, the blood supply is reduced by about 75 to 80%. So the blood supply in this infarcted region of myocardium is very similar to what the myoblasts normally see. So by comparison, they're also very resistant to ischemia. In addition, as you know, you can put a tourniquet on your arm or your leg, and you're not necessarily going to lose your arm or your leg after 4 hours. Whereas if you cut off the blood supply to your myocardium for 4 hours, you're going to have an infarct. So the myoblasts are much more ischemia resistant. We really don't know whether or not the bone marrow-derived cells are. Again, myoblasts are made to be contractile. They naturally develop along a striated muscle pathway. And to some degree, they seem to be able to respond to their environment to develop a cardiac-like phenotype, although they don't convert to cardiocytes. And I'll show you some of our data and their data from a number of other labs that show similar results. So where are we in terms of preclinical data? What we've really done is develop a number of studies where we create, there's a misspelled word there, where we create a cryoinfarction. So we actually freeze a region of the myocardium or ligate a coronary artery at the same time that we take a skeletal muscle biopsy. And this simply shows a piece of skeletal muscle being pulled up to be biopsied. We then expand cells in culture for a couple of weeks, anywhere from 2 to 4 weeks, depending on the number of cells that we need. Prior to transplantation of the cells, we actually do a functional study of the myocardium so that we have a baseline study after our infarct, and then we transplant our cells. After we inject our cells, we wait anywhere from 3 to 6 weeks and do a final study of the myocardium and then harvest the tissue to see if our functional results correspond to engraftment of cells. And one of the things that I'm really excited about, and I talked to Bob about briefly, is the fact that what we're really missing here is a way to show in this final study that we have viable cells in the myocardium. We now are completely dependent upon histology to show us that. So this simply shows alkaline phosphatase staining, purple staining, of a rabbit myocardium in which we've created a coronary artery ligation-induced infarct. You can see there's a transmural scar here in contrast to the rich capillary density in the remainder of the myocardium. But you can see that even though there's a transmural scar, there is vascularity in this region. And that's an important thing to remember. Just like any wound healing process, there's a neoangiogenesis phase that occurs in this infarcted region of myocardium, and there's a blood supply there. The fibroblasts indeed have to have some way to get in to deposit collagen and actually mature the scar. So we take advantage of the vascularity that does exist to transplant our cells in this region. This is an image that many of you probably have seen that was published a number of years ago now by our group, showing our ability to actually, again, create a transmural infarct, in this case by cryoinfarction, so placing a -70-degree cryoprobe on the surface of the heart. You can see again in this H&E stain that there is vascularity in this region of the myocardium. And then this is an island of cells that we were able to obtain after injection of 10 to the 7th cells, 3 times 10 to the 7th cells in this rabbit myocardium. One of the things that you can't really see well in this micrograph, but hopefully you can see at slightly higher magnification, is that the cells orient in a number of directions. You can see cells in this orientation, cells in this orientation, and these cells are actually coming out of the plane of the slide. So the cells seem to get some degree of orientation based on the stress in which they find themselves. And our data now suggests that in fact they're much more dependent on matrix than we originally thought for their orientation. When we stain these samples for myogenin, a skeletal muscle specific transcription factor, you see that the nuclei of these cells were positive. And there's an eosin counter stain here for the surrounding scar. So this is a very cellular region as well. So you see myogenin staining, which is never found in the normal myocardium. And so my initial thought was, great, we've got these skeletal muscle fibers growing in the heart. Cool, how exciting. And we could see this at the gross level. These were on the order of a centimeter in length, and it was pretty impressive. I thought, great, we've got this plug of skeletal muscle. And then we did ultrastructural analysis on frozen serial sections. And what we found was that rather than lining up as skeletal muscle myoblasts normally do and fusing to form multinucleated myotubes, that instead these cells aligned and were connected by intercalated discs. And if you look in this next slide, you can see some striations, but more importantly, you can see these intercalated discs joining these cells. And we could not find any multinucleated cells in this region of the myocardium. We couldn't find myotubes. We could only find myoblasts that would seem to be aligned in this way. What was interesting is Ray Chu's group from Toronto has seen similar results in dog myocardium, and Chaquet's group in France has seen similar results in pig and sheep myocardium. In rat and in mouse, no one gets these results. Instead, what they seem to see is formation of myotubes. And that's one of the Chuck Murray, for example, at the University of Washington at Seattle, sees myotubes in his hearts after he transplants them. Doesn't see long myotubes, but he sees binucleated or to some degree multinucleated cells rather than these intercalated discs. We don't know why. Chuck and I have talked about collaborating, but we haven't done it yet. So we know that myoblasts engraft in high numbers. We can get up to 75% of the scar filled with these cells. However, we also know, and we know that they develop some histologic characteristics consistent with exposure to the myocardium. Although I'm not going to show you these data today, these cells continue to be surrounded by scar. When we look at electrical wave propagation of these cells, what we see is that in fact there is some electrical propagation, but we can't see, if we paste with plunger electrodes, if we paste these cells that have been implanted, we don't see wave propagation to the periphery or to the surrounding myocardium. And conversely, if we paste the surrounding myocardium, although we see contraction of that region, we don't have good evidence of electrical connections. So one of the things that is really interesting to all of us is whether or not we're setting up a proarrhythmic region of the myocardium. And that's something that no one's really got definitive data about yet, although our lab is actively working on that. And we're working to do some modeling with Craig Enriquez to see if, in fact, the cells remain skeletal myoblasts, whether or not they're likely to be more proarrhythmic than if they begin to convert, or conversely. in contrast, my bone marrow-derived cells appear to engraft at relatively low numbers. Even if we put large numbers of the cells in, for a few days we can find large numbers of bone marrow-derived cells. And I apologize for not having a picture. Had a little computer glitch last night. And they seem to develop some histologic characteristics consistent with exposure to the myocardium, but that's only if we put them in normal heart. If we put them in scarred myocardium, we get cells, as I said, that look indistinguishable from fibroblasts or from chondrocytes or adipocytes. And we don't really know yet how to control the phenotype of those cells well enough to use them, in my opinion, for large-scale myocardial repair. In addition, Ray Chu's group has shown that they can get cells, and they showed these data at AHA last year, that their bone marrow-derived stromal cells in rat myocardium, in fact, convert into chondrocytes and adipocytes. So we can get the cells in, they graft, they hang out in the heart, but if they don't do anything, who cares? So what do? Well, we've got data. These data are pressure segment length loops that we've obtained in our infarcted myocardium. So this is a cardiac work loop, the area inside this curve being the work done by the myocardium or work done on that region of the myocardium. These are clockwise loops, indicating that work is actually being done on that region, not by that region. So this is an aneurysmal region of myocardium. It's not doing any active work itself. And we deliver media into that region and then wait 6 weeks, we continue to see a clockwise aneurysmal loop of myocardium. And we obtain these data by implanting 2 ultrasonic microcrystals in the myocardium, measuring the distance between those crystals over a cardiac cycle and measuring left ventricular pressure. In contrast, again, in an aneurysmal heart, when we implant 10 to the 8th myoblast, what we see is that, and this is a rabbit heart, in fact, we convert the work loops from negative to positive. They become counterclockwise, and there's actually an area inside this loop indicating work being done. If we derive the relationship between stroke work and end-diastolic segment length, we get data that look very similar to the Frank-Starling relationship that you normally see in the myocardium. In our control animals, we have a linear relationship. After injury of the myocardium, we typically see a decrement in the slope of this relationship and a shift to the right in the curve. When we transplant myoblasts, we see an improvement in the slope of this line, whereas with sham treatment, we see a continued worsening. These data actually demonstrate that cell number matters. One of the questions that we always got when we published our Nature Medicine paper is, why did only 7 of the 12 animals get better? And initially, we thought it was maybe because of inflammation or maybe because of putting the cells in too early. We really couldn't answer that question. We were speculating. What I think now, based on the experiments that we've done, is that there's a linear relationship between cell number and the reproducibility of engraftment. And so what we found is that if we measure systolic shortening, contractility in the myocardium, sham treatment, so pre-treatment in the blue areas here, post-treatment in the maroon areas, you see that after 10 to the 7th myoblast in a approximately 0.8 centimeter cubed infarct, that we see no improvement in systolic shortening. If we increase the number of cells to 10 to the 8th myoblast, we see a reversal and an improvement or a reversal and a positive stroke systolic shortening in this region. Media continues to get worse, and we've got an outlier here. If we get rid of one animal, basically down here, we get a number that's identical to this. What we've been able to show is that in our Nature Medicine paper, we were at 3 times 10 to the 7th myoblast, and we were right in between these two values, and the relationship we get just as linear as it can be, 0.993 or something like that, showing that below a certain value, or as you decrease the number of cells, you decrease the ability to get reproducible engraftment. So, because I believe a picture's worth 1,000 words, I want to show you this video that some of you saw a minute ago. This is a pig heart. This is an infarcted pig heart. Again, we created a ligation. We ligated a coronary artery, in this case, just proximal to the last diagonal. We created an infarct. You see, again, vascular supply in this region of the myocardium. And then we injected 10 to the 9th cells 4 weeks post-infarct, and I'll show you our wall motion results. So this is an ultrasound image, and we're going to see if I can drive this mouse. Okay, and this is an ultrasound image 1 month post-injury. And if you look at this region of the myocardium, you see there's a dyskinesis. The rest of the heart is pumping very well. This is the infarct region of the myocardium. So then we transplanted cells into this heart. And 2 months later, we measured contractility. And although this is a slightly different view, you can see that when we measure total contractility, there's much better squeeze in the whole myocardium in this region of heart. So these were some of the first data that we got that convinced us that we could, in fact, increase contractility in a synchronous way in the myocardium. So there's not an asynchrony going on. You're not seeing contractility after the rest of the myocardium. You're, in fact, seem to be seeing synchronous contraction. What we found is that in our pigs, time really makes a difference. That at 1 month, we may see an improvement in function, we may not. By 2 months, typically we do. In some animals, it takes as long as 3 months. And that, in fact, suggests that the cells are proliferating in the myocardium, although we don't have definitive data to show that because we can't measure, we can only look at a heart once, unfortunately, by histology. We need good imaging technology to let us actually visualize cells. And that's one of the things that's really limited this field. So, lest you think a cell is a cell is a cell, for comparison, we also injected fibroblasts into the myocardium. And what we found, first of all, this is a scarred region of myocardium. What we found, first of all, is that the fluorescently labeled fibroblasts do not coalesce to form an island of cells in the center of the scar like the myoblasts did. In fact, they're disseminated throughout the scar, and nor do we see any of the histologic characteristics that we saw with the myoblasts. When we look at function, as maybe not as we would have predicted, in fact, in our cryo-injured hearts, we see, again, a linear relationship. After transplantation of myoblasts, we see an improvement in contractility, but after fibroblasts, we see a continued worsening of systolic performance, similar to what we see after sham treatment. And my initial interpretation of these data was, well, we're putting fibroblasts in, we're getting a stiffer region of heart because fibroblasts make collagen for a living, so in fact, we just see a stiffer scar, can't contract as well, no big deal. Well, nature being what it is, that didn't turn out to be the case. We could in fact measure strain in these hearts, so we measured pressure, we constructed pressure-strain relationships. And what we found is, as you would expect in a normal myocardium, as you increase pressure, strain increases exponentially. Whereas in our scarred myocardium, you see we have a very stiff region, as you would expect. When we transplanted fibroblasts or myoblasts, we saw essentially the same degree of improvement in strain or in compliance. And what's turned out to be interesting is if we deliver growth factors, if we give VEGF, if we give FGF, and we increase capillary density in the region, we see essentially the same improvement in compliance, but not in contractility. What the interpretation that I now place on those data is that, in fact, having something living in that region of scar, be it blood vessels or living cells, is actually better for relaxation of the myocardium than having simply collagen matrix there, but it takes a contractile cell to benefit systolic performance. So we know systolic performance improves, and although I didn't show you any global data, we know that it improves globally as well as regionally. And I concentrated primarily on rabbit and pig, but there are data from other laboratories showing improved contractility in rat and in sheep, and also in pig from other from other laboratories. Independent of infarct model, initially most of us were using a cryoinfarction model to simply cut function to 0 and start with a baseline of 0, but more recently we've all moved to, or most of us have moved to more physiologically relevant coronary artery ligation models. We still see the same degree of improvement in contractility if we get the cells in. And it appears fairly independent of the method of assessment. Whether we use sonomicrometry or ultrasound, we still see improvements in contractility after myoblast. We believe that cell type is critical to outcome. We don't see functional improvements with any cells that we've looked at except myoblasts. I think the jury is still out on whether or not bone marrow-derived cells are going to be able to be used in this arena. Right now, we can't control the phenotype of the cells well enough to see an improvement in function. And the outcome appears linearly related to the number of myoblasts, but independent of the baseline function. You know, the bottom line question has been, but is it safe? And the really striking evidence are that none of us have seen arrhythmogenicity in these hearts after transplanting these electrically very different cells into the myocardium. And I think that's actually because the cells are still surrounded by scar, so that they're actually insulated from the surrounding myocardium electrically, and that most of the contractility we see is secondary to mechanical transduction, stretch-induced contractility of that region of the myocardium. And we've started actually making measurements now of contractility. Even though it looks synchronous, there is a several millisecond delay in the contractility that we see in that region versus the remainder of the myocardium. And we're trying to do some much more detailed analysis of that now to see if we can get a handle on whether or not it's electrical activation or mechanical transduction. So how does it work? Does it increase contraction? We believe that it does. We believe that myoblasts do, because we can actually convert negative work loops to positive work loops, and we can't do that with any other cell type. That's our most compelling data, that it actually is contractility versus a change in remodeling of the remainder of the myocardium. There are a number of, it's also possible that we're limiting damage to the myocardium. That is, by putting cells in at a time relatively early after infarct, we've changed the wall stress on the surrounding myocardium so that, in fact, you don't have to hypertrophy the cardiocytes in the surrounding regions. You get less remodeling and, in fact, less dilatation and ultimately failure. And our data are consistent with that as well. And another possibility that we don't really talk about at all is that these cells are maybe just delivering needed growth factors or genes like VEGF or FGF. One of the things that's important to remember is no matter how much cell sorting we do, we're not putting in a pure population of myoblasts. We definitely have fibroblasts in this mixture. And we know that the fibroblasts make FGF. We know that our myoblasts make VEGF. So it's clear that we're also delivering growth factors to this region of myocardium. And in fact, that's one of the reasons to use cells versus genes, I believe, that we're getting a whole milieu of factors delivered. I would say, as I put forth earlier, that repair really depends upon restoring both blood flow and regenerating the injured tissue. And I want to very briefly shift gears and show you some unpublished data where we've actually been able to use myoblasts to deliver VEGF to injured skeletal muscle and improve blood supply to that region of the periphery. Now, we initially published probably 5 or 6 years ago now, data showing that we could deliver VEGF adenovirus to the myocardium and increase capillary density in our scar, but not in normal myocardium. Problem was that in up to 30% of our animals, we got tumors. We got hemangiomas in the region of myocardium where we delivered the growth factor. And thinking very simplistically, I presumed that that was because we had a very high local concentration of this VEGF, and that if in fact we could deliver a lower, more physiologically relevant dose of the virus or VEGF over a prolonged period of time, we might in fact overcome the side effects. And one way to do that was to deliver VEGF protein, which we did, and we saw some increases in capillary density. We got the protein from Genentech and delivered it, and then delivered our cells, and saw increased capillary density and maybe even some improvement in contractility, although those data are pretty sketchy. So instead, we decided to take advantage of the fact that we grow our cells in the dish all day, every day, and that we could in fact infect the cells with VEGF, with an adenovirus making VEGF, and then not have to deliver the virus per se to the myocardium, but deliver the cells making VEGF. So initially we did experiments in the periphery where we took a soleus muscle biopsy, expanded the cells. In the same animal, we created a hind limb ischemia model, or actually this was a hind limb ischemia reperfusion model, and then overexpressed VEGF and injected those cells back into that region of the hind limb. And what you see here, this is Masson's trichrome stain showing the cross-section of a skeletal muscle. You see the scar, you see, and this is a light micrograph. Again, you see muscle fibers, you see the scar and the periphery of the muscle fiber bundle, and you see that our fluorescently labeled, DAPI-labeled myoblasts really move into the periphery of this muscle bundle, and then the cells actually begin migrating in to the muscle, as you would expect if they're going to repair the muscle fibers. But what was really cool was when we looked at capillary density. What we were able to show is that in our control animals, if we measured the capillary density as a percent of the contralateral EDL, after ischemia, we saw about 25% of control. If we sham treat, no increase in capillary density. As a control, we used an empty adenovirus to show that there weren't any changes due to inflammation secondary to our virus. And then we delivered VEGF virus, and we could actually see some increase in capillarity, but not a significant increase. And then we delivered our myoblasts, just plain myoblasts, which were making some level of VEGF and which also contain fibroblasts that make FGF. And we saw actually a greater degree of increase in capillary density than we did even with the VEGF virus. When we gave our VEGF, our myoblasts that were actually expressing VEGF, you could see that we could get back up to about 75% of control. And again, to show you a picture of what that looks like, this is an untreated EDL, and this is or untreated hind limb, and this is after we injected the myoblast expressing the virus, an arteriogram, and you can see the increase in capillarity in this region. So we never saw tumors in these animal models. So we're convinced that we can use these cells to deliver gene products in ways that may overcome some of the side effects of these viruses. So is myoblast transfer going to work in patients? I submit that if you believe USA Today, it already has. This is actually a USA Today special edition published after the French group took these type of data, our data included, and got permission to start a clinical trial in France. And what they actually did in their clinical trial is take patients who were going to receive coronary artery bypass grafting and transplant myoblasts at the time of the coronary artery bypass graft into a region that they said would not be revascularized by the graft, and then measure the contractility and viability of that region, and I don't have the slides, I thought I did. So I'll say, let's see, in patients we know we can grow the myoblasts, and let's see if so the French group then, to date, their surgical results for a combined coronary artery bypass graft and cellular cardiomyoplasty, they've proceeded in 6 patients. There's been 1 death unrelated to cell delivery, although the patient had a mesenteric infarct, and I'm not convinced that we could say that that's not secondary to cell delivery if in fact the cells missed the myocardium and were injected into the ventricle instead. There's been a 3 to 10 month follow-up, and if you look at left ventricular ejection fraction, preoperatively it was on the order of 27%, and there's been about a 13% improvement in EF in all the patients. So this is really a safety trial. So the good news is there has only been 1 death, and that appears to be unrelated to cell delivery. So that 5 patients who've received cells have lived now out to anywhere from 5 months to a year without any adverse events. The problem is that this is a combined trial, and that we really can't say anything about efficacy or about function unless we can transplant these cells in the absence of adjunctive treatments like coronary artery bypass graft. And so there's an investigator-sponsored trial going on at the Thorax Center in Rotterdam. It's a 5-patient trial, and I should say up front that I'm associated with a company, I'm on the scientific advisory board of a company, Bioheart, that's provided the cells for this trial. It's a 5-patient trial. The inclusion criteria, the patients have to be between 18 years of age and 75 years of age, have to have had an infarct at least 4 weeks ago, have an ejection fraction greater than 20%, be able to undergo biopsy. And because this is going to be a single treatment trial, a catheter-based cell delivery, in the absence of any other treatment, you have to have a regional wall thickness of greater than 5 millimeters in the region where you're going to inject the cells, because you really can't afford to perforate the myocardium, and be able to give informed consent. And that trial, 1 patient has received cells, and that patient is doing well 2 months out. My understanding is that 2 patients are to receive cells in the next week or so. And so that trial is well underway. Where do we stand? In Europe, in the US, there's a phase 1 approved trial to deliver myoblasts in patients who receive left ventricular assist devices by diaphragm, and also as an adjunct to coronary artery bypass graft. But again, both of these demand other treatments being done to the myocardium. Now, the advantage of the LVAD trial, in my opinion, far more than the CABBAGE trial, is that at some time, these patients are going to progress to transplant, and that we'll be able to obtain the myocardium and actually look and see if the cells are there. Bioheart expects to receive phase 1 approval in 2001 for an endovascular catheter-based trial in the U.S. that will not be secondary to any other treatment. But I maintain that now is where we really have to stop and think about what we're doing, and that clinical trial design is critical to where this field goes. We've really got to weigh whether it makes more sense to use patients who just had an infarct, where there's less morbidity, and so you're more likely to get a positive outcome, or in patients who have no options, but are therefore at less risk of having negative outcomes from this trial. Because if we begin to deliver these cells in patients who have just had an acute myocardial infarction, without proving first that it's safe, I think we're really setting ourselves up potentially for another gene therapy debacle where we're jumping over the safety hurdle and trying to get secondary efficacy data without doing the appropriate safety trials. There's a huge debate in the field going on. I only bring this up, even at Duke, we have 3 different groups of cardiologists and surgeons, all of which want to go forward on a different subset of patients because they all feel very strongly that a different group is best for the first trial. And they range from patients who just had an infarct to patients who have no option. So where do we think we're going to go? And this year in Europe, we expect there to be a safety efficacy trial initiated. It's going to be a surgical single treatment delivery of cells with no cabbage, no LVAD, just simple delivery of cells. We know the French group is going forward with this, and we know that several companies are. There's also been a bone marrow trial, a bone marrow-derived stromal cell trial initiated in Singapore and in Rotterdam. That's an investigator-sponsored trial that we really didn't expect because there are virtually no preclinical data out there on bone marrow-derived stromal cells with the exception of 2 rat studies. So again, I think that's a little bit premature, but time will tell. We expect an endovascular approach trial. Again, there's an investigator-sponsored trial that's underway, and we expect several companies to begin trials in Europe this year. In the U.S., we expect Diacrin to move into phase 2 trials. We know that there are investigator-sponsored trials that are beginning, and unfortunately, everyone seems to think that anybody can grow a cell and that you can, that any myoblast that you grow is going to work for these trials. And I'm here to tell you, after 10 years of doing this, that a cell is not a cell and that we can't get this to work 100% of the time after 10 years of experience. And every time we have a new surgery fellow in the lab, there's a new learning curve and everything changes. So I'm afraid that we may see some negative data before we see positive data in this field. And we know that there's a bone marrow stem cell safety trial that Osiris is beginning in the U.S. as well. If you don't take anything else home, take home the message that if we do this wrong, we're going to doom this field, and that we're going to set the field back 5 or 6 years, just like gene therapy was set back 5 or 6 years by the unfortunate investigation that resulted in the death of a young man. I don't think I really need to go through this other than to say we've decided to go forward in multiple patient populations. We're going to do both surgical and percutaneous delivery. And initially, although we're focusing on myoblasts, what we've said for a long time is that myoblasts are not the answer, they're an answer. And that we want to continue to develop the answer as we go forward. And initially, we're looking at cells, but ultimately I think we'll be looking at cells in plus or minus genes. And I think I'll just go forward there and say, why is this such a big deal? Well, it's a big deal because there's a huge market out there for this. This is from a document called the Investigator's Guide to the Heart that was published, and the Investor's Guide to the Heart that was published last year. And you can see that cardiac tissue regeneration, where we couldn't even get people to listen to us 3 years ago, is now being viewed as the largest market, $5.3 billion of a $7.3 billion market in the U.S. Well, let's go back one so that I can show you. Go forward one. I really would like to give credit to the people who actually did this work. I've collaborated for a number of years with Don Glauer, who's both a cardiothoracic surgeon and an incredible biomedical engineer who developed the ability to measure regional function in myocardial. I've had a number of surgery fellows who've worked with me, as well as cardiology collaborators, cardiologists Brian Annex, Keith Channon, Stu Russell, and then the people in my lab who really did the work that I talked about today. So I think I'll stop there. Thanks.
Thanks, Doris. Now for questions, discussion.
In relation to the myocardial repair stuff, I was wondering how you envision. You mentioned that these cells, the myoblasts, are more comfortable, or the cardiocytes in the low oxygen environment, that kind of thing. Do you envision that? How do you envision it remodeling? So do you expect them to stay in a low oxygen environment and just be completely different tissue, or is it going to end up being normal cardiac tissue, or?
Well, I think the only way to answer that is to do some long-term experiments, and as you know, those are very expensive and we haven't done them yet. But I think that's actually an important safety consideration as well, because if we really view this as something that's going to work after an acute myocardial infarction, these cells are going to have to be able to hang around for years, and or we're going to have to be able to redeliver them. And there are some data from the French, some of my French colleagues in rat, who've been able to show that these cells can hang around for 15 months and still survive and seem to improve contractility. But one of the things that's really kind of interesting is that a number of people have obtained data where they deliver these myoblasts, they see improvements in contractility, and then they harvest the hearts and they look for the cells and they can't find the cells. And they don't know why. And so we don't know if the cells actually are there for a while and then go away, or what. Do I think these cells are going to ever become mature cardiocytes? I think that completely depends on what we do to them in vitro. That if we push them too far along a differentiation pathway in vitro, no. They're never going to become cardiocytes. Do I think they have the potential? Yes, but only in the right conditions. We're doing some co-culture experiments now to try to understand some of the changes that occur. And what we've found is that we can, by culturing these cells under appropriate either electrical stimulation, I believe that these changes are superimposition of electrical, mechanical, and chemical changes, and that any one alone is not going to give us the effect that we need. So by doing some co-culture experiments and some electrical stimulation experiments, we're beginning to get data that show we can get cardiac transcription factors turned on in these cells under appropriate co-culture conditions. And that if we take conditioned media from the co-cultured cells, we can begin to affect myoblasts alone. So it's likely that we're going to be able to ultimately find the factors that may play a role in this, but we are a long way from there. We've tried transfecting cardiac transcription factors into these cells in vitro and then trying to push them down a cardiac pathway, and boy, is that hard. It just hasn't worked well. I really think nature does it much better than we do, and even under the co-culture conditions. And I should say that by co-culture, I don't just mean cardiocytes, I mean cardiocytes and cardiac fibroblasts.
Let me piggyback on that, because early on when we were comparing cell types, one of the advantages of skeletal myoblasts was that it was a contractile phenotype, and you said partial cardiac. What did you mean by saying partial cardiac phenotype, which was in addition to it being contractile?
What I mean by that is if the cells start expressing cardiac gap junctions, and also I should say, and I didn't say this, we've chosen to derive cells from oxidated skeletal muscle. And if you think about skeletal muscle, it's a continuum. There's glycolytic muscle, there's oxidative muscle, and then there's the heart, which is the ultimate oxidative muscle. And I think by choosing cells at the oxidative end of the continuum, we've chosen cells that already express beta-myosin heavy chain, for example, and express some of the same contractile proteins that ventricular cells already express. Whereas at the glycolytic end of the continuum, they don't express the same contractile proteins.
In grafting of these cells, what role does the extracellular matrix play, and what are they adhering to? Do they remodel that? What extracellular matrix are they interacting with when you're engrafting these cells, and do they remodel that extracellular matrix?
I'm speculating. We know that as the scar matures, that we go from type 3 to type 1 collagen in these hearts. What we don't know is whether or not engrafting cells makes a difference in that. We've just started barely measuring MMPs. One of the things that I've been really interested in is whether or not we can overexpress some of the MMPs in our cells and put them in a very mature scar, and put them in the periphery of a very mature scar and see if we could actually get them to migrate in better. We know very little about the matrix, and that's a real limitation, but that's just not been something we've had the resources to do. It's something I'd be very interested in knowing a lot more about, because until we understand the environment in which these cells find themselves, we're really working in a black box. I think your question's right on, and I just don't know.
Sandy?
Well, you mentioned that if you believe that myoblasts are an answer, so in the search for the answer, are you going to work with genetically engineered cells or other cell types?
Sure. The question was, are we going to work with other cell types or genetically engineered cells? I really like to take the approach that simpler is better, especially if we're thinking about doing this in patients, because it's a whole lot easier to get a myoblast and not have to do a whole lot to that cell, or to get some bone marrow and not have to do a whole lot to those cells, because less can go wrong. On the other hand, if we can't provide a long-term solution without doing something to those cells, then I support doing something to those cells. Dr. Niram and I were talking about the fact that allogeneic cells obviously would provide the opportunity for tissue banking and for having an off-the-shelf product and much more reproducible batch release criteria if you're talking about a product. On the other hand, when you have to then deliver immunosuppressive reagents to people who are already very sick and who already have compromised renal function because they have heart disease, then you're looking at a significant downside. So we've chosen to initially go with autologous myoblasts and to start working with other mixtures of cells and other cell populations to see if they make sense.